I want to thank everybody for joining us for today's session entitled Digitalization of Therapeutic Antibody Discovery and Development, brought to you by Bionamics. We can go ahead to the next slide, please, and we'll do a couple of quick introductions before we get kicked off. If you haven't attended any of the Asterix partner webcast series that we've been running over the course of the past year or so, we want to welcome everybody to this. I want to give you a quick overview of who Asterix is, and then we will introduce our speaker for today and let them pick it up from there. So real quick, in terms of the company, Asterix is a company that specializes in professional services in the life sciences space. We do highly specialized services, process technology, as well as outsourced services to fundamentally transform how science-based businesses operate. And we do this in an effort to improve scientific outcomes and the lives of people everywhere. A couple of quick statistics about us. We were established in 1995, or privately held, but we originated as an IT division of a company called APBI, which was a life science research organization. We operate from 7 offices here in the United States and 1 in beautiful Costa Rica, and we have our headquarters in Red Bank, New Jersey. We are a market leader in dedicated digital transformation services and expert outsourced services for the scientific community. The type of companies that we support are going to be everybody from Fortune 1000 life science enterprises to chemical, CPG companies, government research institutions, generally speaking, those with large and fast-growing IT and outsourced and compliance needs. We can go to the next slide, please. In terms of the services, so the different services that we deliver, we like to say that we provide a suite of services that span the entire life cycle of scientific data systems needs. And this is, from the very beginning, we come in and help companies develop a sense of what their architecture is going to be. We help them through the business process analysis of, what new technology is going to mean for their organization. And this can be everything from helping them plan, what is the technology stack going to look like, what it's going to sit on top of, is this cloud-based, hybrid, et cetera. And then we can help get into the actual selection of different applications and services that will support that architecture and that business process. From there, we can help with development implementation if need be. And then when that's finished, we can assist with the computer systems validation to make sure everything meets regulatory guidance. After that, we provide a host of on-demand services from everything from helping support, the overall architecture to supporting unique applications, et cetera. And then we can even provide specialized scientific and technical staffing on demand. So if you need folks in your lab or if you need folks to help support those systems, we can provide those resources to you. So that's a little bit about us. And I want to switch gears and introduce our speaker for today and let him take it from here. So Anders is going to be speaking. Anders Carlsson, he's the CEO of Bionamics, which is out of Lund, Sweden. Anders is a researcher specializing in data analysis and structure. He founded Bionamics in reaction to years of working as a data analytics consultant at various biopharmaceutical companies. He observed some of the challenges with navigating and analyzing the data generated in antibody discovery campaigns, which led to the solution to meet some of those challenges head on. So I'll introduce Anders and let you take it from here, and then Anders, I'll come back on at the end.
Great. Thank you, Kevin. It's my pleasure to be here today and tell you a little bit about Bionamics and the work that we've done. But I thought I'll start off by introducing therapeutic antibodies. I guess most of you interested are already familiar, but looking at the top 10 list of selling prescription drugs, 6 of them today are actually therapeutic antibodies, and that's out of a total of about 80 antibodies that have been approved by the FDA for treatment of various conditions, including cancer and autoimmune disease. Looking in the pipeline, there's about 600 of them currently in clinical trials. So the 2019 market size of about $150 billion is expected to double by 2025. Now, to the right, you see a cartoon of a typical antibody structure. So an antibody is composed of four protein chains, 2 heavy chains that are depicted in blue, and a light chain, or 2 light chains in green here. So the darker shades of blue and green represent the constant regions. So there are a limited number of different sequences that compose these regions in any given antibody. Now the regions in yellow here are the variable regions, and those are the regions that are responsible for actually interacting with and binding to your antigen. So the protein sequence in these regions highly variable, and is what's responsible for the fact that antibodies can bind to almost anything, and in particular these CDR regions that actually do the binding in most cases. So obviously, like any drug discovery development project, creating a therapeutic antibody is a long process with tons of different steps, each generating data of different kinds and in different amounts across this road, from just having identified a target to having a drug candidate. So now integrating all this data and tracing it and analyzing it in a powerful way is obviously a very complex thing. So I highlighted here a few of the steps that are relevant in such a process. First of all, you need to start perhaps your project by generating candidate binders against your target molecule, and that could be done either by doing animal immunization, so injecting your target into mice or rats or other animals, or you could use recombinant technologies like molecular libraries and use phage display or other display systems to start panning for your candidates. Once you've done that, you need to start ranking those candidates and do hit selection. So depending on their binding properties to your targets, you need to sequence them and then keep characterizing them functionally, including what kind of epitope are they binding on your targets, do they have blocking properties, or are they agonists, antagonists, and so on. Further, you may want to convert that binder into different antibody formats. So you may, if you come from a recombinant setting, you may have a single chain antibody or a Fab fragment, and you want to put that into one of multiple frameworks. And if you came from an animal immunization, you will need to do a humanization as well. So to move the antibody into a human framework. And of course, there are many other properties of these molecules aside from their target binding. So you want to avoid aggregation, and these sequences may have motifs that introduce post-translational modifications that you want to engineer away, and so on. After that, you have to optimize production protocols and purification protocols and so on. And today, the new black, perhaps in antibody development, is to create these bispecific antibodies or even multi-specific antibodies, so that could bind to more than one target. So there certainly are a lot of decisions made in this process, and that the data that needs to be captured, and that is the kind of the background that we or the context that we started Bionamics in. So myself and my colleague Morten, we have worked for a number of years doing various consulting within biopharma and other industries with a focus on data analysis. And we've seen firsthand how frustrated the folks at these companies are with managing and analyzing the data. So a little bit about our backgrounds. I'm a PhD in immunotechnology from Lund in the south of Sweden, and I've done postdocs here in the US at Scripps and USC before going into more of this data analysis consulting. In contrast, my colleague is actually a physicist who has worked with theoretical physics, but has since transitioned into bioinformatics, as I said, but also worked at various startup companies as a more traditional software engineer. So we thought we had a pretty interesting skill set, the two of us, to try to take this problem on. So what we kind of asked ourselves was that how would a software platform work if antibody researchers could essentially wish for anything? And so what we've done over the last 2 years is to sit down and have a close collaboration with a number of antibody R&D groups, both private and from academia. And our team's grown to now to 12 people. And what we've realized during these years is that it is an incredibly hard problem, first of all. And it has required us to rethink things, and we've gone through several iterations of this platform. But we're now very happy to be at a point where we have a platform that's been incredibly well received by our customers. So some of the observations that we made is that, perhaps most importantly, that scientific data and data in like electronic lab notebooks and LIMS systems are not actually separate things. It makes sense in a way to build different systems to store this data, and you can certainly do integration between them. But ideally, we feel that the data should be generated together, regardless of what type of data it is, and it should be stored together and also accessed together. We further learned that requirements on the ease of use is very high, and that a single software should be able to do both very advanced analytics for a power user, while at the same time being accessible for example, executives who wants to go in once every 3 months and do a quick search for something, and without really relying on any formal introduction or training on the software. So that's a difficult equation, but it's I think one that's important to solve. And perhaps most importantly, what we really want to do is start eliminating these error-prone manual steps, which obviously can be disastrous if they get introduced early on here in a project like this. So, since considering where we are, I thought we perhaps don't need to talk that much about why digitalization is important, but so we can focus on why it's hard instead. And I think what we realized that interactions between humans is pretty powerful, right? So when you're talking to someone else, we're really good at making ourselves understood, even if we are being sloppy or incomplete when we say things. The power of a follow-up question and clarifications and confirmations is extremely high. So if you contrast that to the setting where you actually need to document for posterity, where incomplete and inexact information will result in loss of information. So a user uploading and describing data into a system, time goes, the user may leave his or her job, and then a year later, some other user wants to access that data. You're kind of left to your own devices at that point, in contrast to having someone to talk to. So I'm going to give you a quick example. So you can imagine this being 3 very simple Excel files. The first one to the left describes 2 different productions of 6 different antibody clones. So clone 1 through 6 was first produced on May 7th with some concentration, and then a week later 4 of them were produced again. And then we have a file with some ELISA data that mapped to these clones. And that was done May 9th. And then a week later or so, another ELISA was run on 4 of those clones. So you can imagine two users sitting at a computer with an Excel sheet, and they can kind of piece this together, right? The May 9th, that's obviously this production. And then May 17th, those 4 clones, they match exactly the ones produced here. So we can kind of piece this together again without problems. But imagine instead uploading this to a database where the full context isn't really known. It actually is pretty hard to guarantee that these ELISA values don't belong to the first production and not the other, right? So it's not really information complete at this time. So you could avoid this by for example, registering each of these samples, giving them names and IDs, and then make sure that when you register the ELISA data, you don't just reference the clone ID, but the actual purification sample. And that works well. But if you want to start introducing these kinds of you want to enforce these patterns into a system, what you get on the flip side is that you get a much more rigid system. A system that will kind of take control away from the users, and that, as we'll see, is also problematic. So, for example, let's say I never did the second production here, and I, as a user, I knew that, well, we'll never produce these clones again. Why do I need to register and give these samples labels? Can I just map my ELISA data to the clones? That's what I'm interested in. So we actually feel pretty strongly about that. We should be able to allow users to do things this way instead. And if you don't, you'll end up with a system that's too rigid, and that will stop making sense to them, and they'll stop using it. Yeah, so we have to realize that there are limitations in what could be documented. We can't capture all data. So a silly example is that, you know, why not register the temperature and moisture in every cube inch in the lab during the time of your experiment? So it's obviously not doable, but it's not, you wouldn't consider it information lost really anyways. So you can mitigate this by prioritizing using a system that is designed to solve specific problems, and that promotes the capture of relevant data. It's also true that documentation could be a significant increase in the workload, especially if the system isn't really well adapted or poorly designed for your question. So instead, a good system should actually reduce the total workload, both short and long term. And again, compliance will suffer if it's too cumbersome or doesn't make sense. And so, as I've said, having this in mind will increase compliance, but you can further do so, we believe, by having some kind of immediate payoff when you use the system, right? So it's not just for future benefit that you want to do your documentation, but you can have an incentive that if you actually save time right here, right now, when you use the system, you'll end up having users that use it constantly. Okay, so a couple of other points that's kind of gone into our philosophy around how we design the system is that we actually want to minimize the amount of predefined workflows that exist in the system, and that is while realizing that workflows can be really powerful. But the context that we're in right now with early phase discoveries, it's also true that these are so dynamic, the workflows. They change every time. You know, you have some new experimental parameter, so the workflows end up needing to be very flexible, and then they kind of start losing their point. So we like to design it as modules that do a fixed thing, but users can kind of string them together in a real-time workflow, in a sense. We also think that properties on so the things that you do register in the platform should also be dynamic. So to avoid this avoid the risk of needing to do artificial data constructs, a little bit like what we talked with those ELISA files. We can rely on power users, you know, experts among our customers, but we need not require programming or database knowledge. So users are typically not interested in writing traditional database queries. So instead, we need to have very intuitive search functionality and a way of browsing your data that makes sense. And finally, we do a lot of automatic automatically create a lot of different relations between the data points in the system. And I'll just briefly show you a schematic of that. So the data points, or items as we call them in our system, they are deeply connected through like this network of relations. And both the items and the relations between them are designed to as closely as possible map what the real nature is of the things that they represent. So, for example, molecules and samples and plates. We try to have a data structure that's really reminiscent of what it actually looks like in nature. And all these things, these items and the relations, again, are dynamically created when you do take actions in the platform. And also the types of questions that the researchers want to ask their data is incredibly important when designing this. So, and that could be like, where was the original version of the VHDL sequences in this antibody identified? And that could be, you know, from immunizations in a particular animal, or it could be during a panning of a library against a target. So that's the type of data that's then captured in these items and relations in an automatic way. Or it could be which purified anti-PD-L1 IgG4 antibodies. That works well in facts do we have in our system. And that's also the type of question that's really hard to formulate in a traditional query. So instead we like this combination of having like a simple, more Google-like search, and then powerful ways to navigate the system. So I actually thought I spent a few minutes just hopping into the platform, and so we're now live in our system, and obviously I'm not going to take you through the whole thing, but I thought I'd show you what it typically looks like. So what we're looking at here is a list of all the VH and VL sequences that are registered in this platform. So a VHVL again is a unique combination of a VH variable sequence with a VL variable sequence. So the binding domains of an antibody. So we can click any one of these, get some more information about it, including obviously the sequences and again the relations. So this VHVL has relations to the individual VH and VL sequences. We can see what targets it's selected against, what antibodies that have this as a subsequence in them, what the DNA is translated for, if any of the antibodies that use this VHVL has been purified, and so on. And maybe to make it even more clear, we can look at some of the assay data that's been registered in this just in this demo data set. Further, so these are the VHVL molecules inside those samples. Well, give me those, and I could keep going and navigating this complete tree in parallel. But for example, I might now be interested, since these are my hit clones, well, let's tag the VHVL sequences with something too. Well, I want my hits tag on the molecule as well. And I further want to now get them into a full IgG format. So I can go to any of my predefined antibody templates, and these are easy to create with the tool that's included in the platform. I can say, well, I want them into my IgG format. I just refine them again here. Select them all, and just by clicking register, I will now generate a new bunch of antibodies. So we'll go to my antibody list, and we'll see that these were just now created. You can click any one of them, get their full sequences, and you'll see that they now is included in this big network of relations. So I can see, well, what other antibodies are sharing this HVL. Well, yes, there is a single chain that was registered yesterday. They all have the same target. This was a template that generated the antibody. And another step that we perhaps should have done first is to look at the VHVLs that we see that we aligned, but we can skip that at this time. So obviously there are tons of tools. This is just a little bit of a quick intro, but to summarize, I think that we've learned that the requirements on a modern and efficient system like this is extremely high, as it should be, right? People, I think, people becoming scientists today are used to a certain level of user experience level in most tools, and I think the demands are increasing on usability and overall experience. The system should also be lean and to the point, right? So we users absolutely hate these kind of artificial steps that they need to take just to get the data in there. And to summarize, yes, this is a platform that's fully designed for antibody discovery and development. But as I'm sure you realize, most of these concepts, the general pattern in how we solve things is very general, and will be applied across other segments of biotech in the future. So thank you very much.
Anders, can you hear me okay?
Yes.
All right, great. I wasn't sure there because I had originally dropped some audio there, but I am back. Cool. So I appreciate the presentation. That was awesome. I'm going to go ahead and get the recording stopped, and I wanted to remind folks who are on